Hello, and welcome back to Psych 300 Brain and Behavior. We'll be discussing Chapter 5, the Synapse, which will talk about synaptic communication. We just previously were talking about communication within the central nervous system, talking about how it's an electrical communication or language within the neurons. And we hinted a little bit how part of the communication between neurons at the synapse is chemical in nature. And so this chapter is going to focus a great deal on the synapse and what that communication system, how it functions, and some of the characteristics. So we'll first talk about the discovery that chemicals were the basis of the communication at the synapse. We'll talk a little bit about the anatomy of the synapse, what it looks like, where it's located, and its effects on transmission of information. We'll talk a little bit about neural transmission, how we get that signal across there, and then we'll talk about different types of neurotransmitters and their role. Then we'll focus and talk about different types of receptors. So the neurotransmitter is the chemical, the receptor is where it binds and has its effect on that next neuron. Those receptors are an important component. That will set us up to look at something that I think is very interesting, almost one of my favorite kind of sections of this class to talk about, is synaptic learning. We'll get to see that changes in the synapse is really the basis of all of our memories. So an ability to remember what you had for breakfast, to that very first memory of what maybe your 6th or 10th birthday party was like, your grandma's face or grandfather's face, those kinds of things that are the memories that stay with you for life, are really can be tied down or depend on that synaptic learning, changes in the synapse. And so we'll see an animal model of this and use it to model a very simple form of learning, but recognizing that this can be related to all forms of learning and memory, including what you're learning in this class. So let's go ahead and get started. We had started our last chapter talking about Galvani and his observation of the frog legs that were twitching in the storm, rolling in and seeing that maybe there was electrical signal that was important for the communication of the nervous system. We'll start this with another story about Otto Lowy. Otto Lowy was a physiologist, and he discovered through an experiment that chemicals are another important dimension of communication in the central nervous system. And the story surrounding his discovery, again, this has been retold in several neuroanatomy or neuroscience history books. The story goes that he often had dreams, and those dreams inspired him to come up with an experiment. And so one night he was trying to figure out how he was aware of the work of Galvani, but he didn't think that electricity was the whole thing. He was interested in this idea that maybe chemicals were an important basis for communication, and he wanted to try to find a way to, he was thinking very hard to come up with a way to test this experimentally, to demonstrate evidence for a chemical signal being important. And so one night he went to bed and he had this dream, and it was very vivid, and he woke up from it and he said, that's it. That's the way we got to test this. This will let us know if there's a chemical signal that's important. And he woke up the next morning and he forgot it. And the next night he went to sleep again, but this time what he did is he put a notepad and a pencil next to his bed. And so by doing that, he was hoping he might have the same dream, because he spent that day thinking about ways to try to test, is this a chemical signal that's the communication of the central nervous system, is this a component? And he woke up in the middle of the night, had the dream, and he wrote down what was in the dream. He woke up again, he didn't remember it, but he remembered he had the paper, he looked over the paper, and there was the experiment. And above here, what we have is a simple diagram of what his experiment was. So he recorded from a beating heart. So these are actually two frog hearts that he had in jars. He was able to keep alive, and they would beat. And he was able to apply a small electrical stimulation to one of the hearts, the vagus nerve, which the vagus nerve in a frog heart and human heart causes the heart to beat slower. So if you stimulate the vagus nerve, which you can kind of see here, this is also in your textbook too, so if you wanted to follow along, you could. There's this little yellow line here that represents the vagus nerve. And if you stimulated that, that would cause the heart to slow down and its rate of beating. Now, what he would do then is take some of the fluid, so he would cause the stimulation to slow down. What would happen is, with this condition, he would have two jars connected, and he would record the rate of firing of the heart in another jar. And the fluid surrounding this first heart would flow across here, and if it was a chemical signal, by stimulating this vagus nerve, that fluid should then gradually come over here, diffuse across this connection, and then impact the behavior of the second heart. And what he found is that the fluid, when it was transferred, it decreased the rate of firing. So he was able to tell that this fluid, or the chemicals here that were released because of stimulating this vagus nerve, caused the second heart to slow down, decrease in its firing rate. So this was evidence that there was some chemical signal. Now he was able to, the great thing is he was able to take this fluid and then look for what chemicals were in it. And he discovered acetylcholine. So when acetylcholine is applied to cardiac tissue, it causes it to contract much slower. It slows the contraction. By the way, acetylcholine is a major neurotransmitter in the central nervous system and also released at muscles in the body. Acetylcholine has a very different effect on the muscles of the body, the skeletal muscles, that causes them to contract. So it's an interesting kind of lesson that one neurotransmitter may have different effects depending on where it's placed. In the frog heart and human hearts, when we place acetylcholine, it actually slows down the heart. But when we place it onto skeletal tissue or onto neurons, it can cause them to increase their firing. So there's some interesting aspects, and we'll talk more about neurotransmitters later on, and especially in the drugs and behavior lecture that comes after this one. Well, he went on to stimulate another nerve that comes into the heart called the accelerator nerve. And he found very similar effects, with the exception they were completely opposite. So he would stimulate the accelerator nerve in this heart, and it would cause the heart to speed up in its firing rate, or heartbeats that would occur. And as that fluid was transferred over, we'd see that the second heart would start to speed its beating as well. And the chemical, after he took that fluid and isolated it, was the chemical in there was epinephrine. So epinephrine caused the heart to fire faster or beat faster, whereas acetylcholine caused the heart to beat slower. And this was the first piece of evidence that chemicals were an important factor in the communication of the central nervous system. And it all came from a dream that Otto Loewy had the foresight to write down that second night. At least that's the way the story goes. The experiment won Otto Loewy the Nobel Prize in 1936 and has served as an important guiding point for our research into understanding the chemical basis of in the nervous system. Now, what's happening is we have synapses. So last class period we talked about the neuron and how the axon synapses on another neuron. We talked about those excitatory and inhibitory postsynaptic potentials and stimulating these different neurons. So what is the synapse? And let's talk a little bit in terms of structural characteristics. What are some of the important players at the synapse? First and foremost, we have the axon. That's that portion of the neuron that leads away from the cell body or soma. And from there, we have the axon that's going to be wrapped in myelin, and we have those nodes of Ranvier, and then that comes down to this presynaptic terminal. So here's the axon. It's leading down to this presynaptic terminal. So all this is the presynaptic terminal. Within the presynaptic terminal, we have vesicles that are filled with neurotransmitter. We see some are still connected up here to some of the microtubules that they followed after the neurotransmitter was synthesized. You can have some of them that are stored together in these granules. And then what we see in addition is we have this presynaptic membrane here, and one of these vesicles has docked, and we can see that it's releasing neurotransmitter into the synaptic cleft, or that break, a very, very small space, tiny space between this neuron and the next neuron. So as these neurotransmitters are being released, they then diffuse across very quickly the synaptic cleft, and they bind postsynaptically with the different receptors that are located on this postsynaptic membrane that's located here, which in our example is a dendrite. So this is a dendritic spine. Here's the dendrite from this neuron, and then this is a spine or a little bump that comes up off of it that this neuron is synapsing onto. So it kind of fits around it like a cup, and very close relationship between these two. And then this will cause the release of this chemical will produce potentially an excitatory postsynaptic potential or an inhibitory postsynaptic potential, depending on what receptors are located postsynaptically, what neurotransmitter is being released presynaptically, and a variety of other factors we'll see. So basically you have this structure that makes up a synapse. Now, synapses can vary a great deal in the way they look, their structure, and from this, researchers have discovered that we can examine excitatory synapses and inhibitory synapses. So in an excitatory synapse, if that neuron fires, we will be producing excitatory postsynaptic potentials. And there are several structural features that are unique to excitatory synapses. First, excitatory synapses tend to be located on the dendrites. So if you were to look for excitatory synapses, you want to be looking at where those dendrites are. This isn't exclusive. It's in general, we see that a lot of excitatory synapses occur on dendrites. So here is an example of a excitatory synapse, and it's occurring here on the dendrite. We also see presynaptically there are a lot of round vesicles. So we see the vesicles are round, and there's a dense material at the presynaptic membrane. So you see a lot of content is being released. You have these round vesicles, and there's a very dense amount of material on the presynaptic membrane. There's also a large active zone postsynaptically. So we see that this is a large active zone where there's a lot of receptors connected there, and there's a lot of neurotransmitter that's being released. So those are characteristics that you would see in an excitatory synapse. They're primarily located on dendrites. Now, we see that inhibitory synapses look different. They, on the other hand, have oval vesicles, or flattened vesicles here presynaptically. They also tend to occur on the cell body. So in general, you tend to see more inhibitory synapses on the cell body. We're going to see that you can see synapses across a wide range of areas. But if we were to say where excitatory and inhibitory are occurring, in general, excitatory occurs on the dendrites, and inhibitory tends to occur on the cell body. Now, we see that we have these flat vesicles here, as well as a sparse presynaptic membrane where there aren't a lot of active kinds of material being released there. So it's a little bit more sparse. We see the synaptic cleft is a little bit more narrow, so it's narrow relative to what we saw with the excitatory synapse, and it's a smaller active zone or a more sparse material postsynaptically. So we see that this is a little bit less smaller active zone than we saw with our excitatory synapses. Here we see that there's a wider cleft with our excitatory, a much larger active zone here presynaptically and postsynaptically. With the inhibitory, we see that there is a narrow cleft, smaller, sparse active zone presynaptically and postsynaptically. So with this tool, understanding this, now we can go in and see where all are these synapses occurring. And we can talk about many different types of synapses. And your book reviews these, and I'll go through these briefly. But what I really want you to understand and take away from this is the idea that depending on where the synapse occurs will differentially influence the information flow through this neuron. Remember, dendrites collect, the cell body integrates, and the axon communicates it on to another area. Could be another neuron, could be another part of the body, it just depends. It could be a muscle, it could be a capillary. And depending on the type of synapse you have, it influences that flow of information. So here we have dendrodendritic is a type of synapse, and this is where two dendrites may come very close together, and if the membrane potential is a certain way, you may see some neurotransmitter interfering or influencing the function of the dendrite. This is a great big influence on flow of information. It may facilitate or something along those lines, but this isn't a very prominent kind of synapse. But it's a type of synapse you may see, and it plays a relatively minor role in this. Axodendritic, we can see this is what we were just kind of talking about with our synapses, where there's an axon and it synapses on a dendrite. These can be mostly excitatory, but you will see some inhibitory examples of this. Again, this is just a small synapse here, so it's going to be a relatively small player in generating an action potential down this axon. So both dendrodendritic and axodendritic play a relatively small role in the flow of information across the neuron. The next synapse is axo-extracellular. This is where an axon can release neurotransmitters into this extracellular space. And that may have a larger impact because these neurotransmitters might hit a much larger area than just what's being hit by axodendritic. So this could be a larger impact. This could cause more EPSPs or potentially IPSPs and either tune up and prepare for an action potential, and or it could cause IPSPs and inhibit the action potential, or EPSPs and cause an excitatory potential. So axo-extracellular can hit a wider area, maybe two to three times larger than what just a simple axodendritic would be able to influence. The next, and now we're starting to get into a zone where we're going to see principally more inhibitory neurons. Axosomatic is where we have a synapse on the cell body, or the soma, hence axosomatic. This is where a axon is going to synapse on the cell body. And these are going to be more inhibitory. And so you can kind of think of each one of these little axodendritic having its initial function, and then it's starting to culminate where it looks like we're going to get an action potential, but all of a sudden here you have this inhibitory connection, shuts down all that processing. So already axosomatic is going to be able to cancel out potentially all these tiny little axodendritic or axo-extracellular kinds of synapses that are occurring and shut down that action potential, preventing it from happening. So the axosomatic is stronger in function. In addition to axosomatic, we have axosynaptic. This is where an axon can synapse and modulate the effects of that axosynaptic or that other type of synapse that's occurring. This can either be excitatory or inhibitory. And this can be a little bit confusing. So imagine that we have an axosynaptic, we're collecting all that information, we're getting ready to fire an action potential, and we have an axosynaptic that tries to turn off that action potential and inhibits it. But what happens is we have this other source of information. You can imagine we're collecting information from senses of touch. Across here, we get some type of information that says we should prevent some kind of behavior, some kind of response. That's what the axosynaptic. But in fact, we need to keep that kind of response. So now this axosynaptic can cancel out that canceling response, and that will then allow the response to continue down through the axon. So we thought we wanted to cancel that, but because it's really important, then we don't. You can kind of think, we're getting this information that our hand's getting hot, and we want to release that, and so this is trying to communicate we need to release that, and then all of a sudden we decide not to, and we cancel out that signal. So that's a good example of why we may want to cancel that information, but at some point that canceling of the information might be problematic, and we need to change the course. So that's why we might have this axosynaptic kind of synapse. Our next type of synapse is axoaxonic. This is where an axon synapses on another axon. This is very powerful. It's already made it through all the dendrites and all those axodendritic, axoextracellular, axosomatic, axosynaptic. It's made it through all these stages, and it's cleared the axon hillock. And so that action potential is now traveling down the axon, and you can have one simple synapse here shuts everything off. So this is going to be exclusively inhibitory, and it turns that signal off. So this is very large. It can really turn off and significantly influence the processing of information. So axoaxonic is inhibitory, and it can turn or negate all the information processing that's occurred up here in the dendrites and the cell body. So axoaxonic is a huge inhibitory signal that just shuts everything off. Finally, we can have axosecretory. This is where this neuron may be involved in some kind of response, stress response to the body. And what happens is gathering information up here in the dendrites and processing information here, whether we should become stressed with, whether we should be frightened or not. What happens is if it makes it through all these stages, it can release neurotransmitter into the capillaries, and then that can spread throughout the entire central nervous system. It can activate other systems throughout the body and initiate a stress response. So this can have a huge polarizing effect in recruiting the entire central nervous system, increasing arousal, or preparing the body to fight. So axosecretory can have a dramatic impact on the entire functioning of the organism. So you could argue that this may be the largest. This may be the most influential, because it's not just at this simple neuron, but when it releases neurotransmitter or hormones into the circulatory system, that can influence the function of the entire central nervous system. So this could be a very large effect. So given that, how does this neurotransmission take place? And so we're going to discuss some of the stages in neurotransmission. We're actually going to come back to this in our next chapter, because we're going to see that these are many of the places where drugs can have their effects. And this many levels or stages of neurotransmission are also important, because this is where learning can modify some of the, we'll see towards the end of this section, can be modified in order to change the rate or the efficiency of communicating information across the synapse. So our first step in neurotransmission is synthesis and storage of the neurotransmitter. So either we have to activate the DNA, we talked about that, and from the activated DNA we have to then transcribe that into messenger RNA. Messenger RNA then translates into protein via the ribosomes. Those ribosomes are packaged up at the proteins are packaged up at the Golgi apparatus and the vesicles. Those vesicles can then be communicated down here via the microtubules and then removed and stored someplace. So we've made those there, and those are some neurotransmitters are made that way. Other neurotransmitters are actually made on site from chemicals that are local around the synapses. So they're absorbed and they're synthesized, and then they have to be packaged up locally here in the synapse. If you don't package them up, there are a lot of enzymes that are present inside of the synapse, and they can break down those neurotransmitters. So it's important to keep everything packaged so you can have a controlled release of the neurotransmitter when it's required. So again, you have these precursor chemicals that are found outside of the neurotransmitter, and they can be used to make neurotransmitters on site, or we can have them neurotransmitters made from activation of the DNA. So these two things are important in synthesis and storage stage of neurotransmission. So we need to make the neurotransmitters, and then we need to store them. After the neurotransmitters have been made and stored, then we need to release them. And what happens is a action potential is traveling down the axon. When it reaches the presynaptic membrane here, or the terminal button presynaptic membrane is located here, what happens is it activates these calcium channels here that are located around this membrane. Those calcium channels become activated, calcium enters in, and then it forms a compound with calmodulin, and that complex then can either remove the neurotransmitters from the microtubules, or it can cause that vesicle to dock here presynaptically, and causing that bubble to merge with the presynaptic membrane, and that merging process then causes the neurotransmitter to be released into the synaptic cleft. So as that neurotransmitter travels down, it activates these voltage-sensitive calcium channels here. Calcium enters, and when it enters, it can either tear the vesicles away from these microtubules, or it can actually cause the vesicles to dock, almost like a bubble landing on a surface, and then as that bubble's there, that causes a pore to open up, and then all the neurotransmitter that was in that vesicle will empty out into the synaptic cleft. So here we see that process occurring, where action potential travels down, it opens up these calcium channels, calcium flows in, and then binds with calmodulin. That calmodulin can either tear off some of the vesicles from the microtubules, or if there are already vesicles here, it can cause it to dock, and then that causes that hole to open up, and then the neurotransmitter starts to fall out. So this is the release of the neurotransmitter. After it's released, what happens? Well, we have to activate the receptors. And so receptor activation involves the neurotransmitter diffusing across that synaptic cleft and binding here postsynaptically. And we've got an example here where it's an axodendritic kind of synapse. So this axon is releasing neurotransmitter, and we have a dendritic spine here, and it crosses the cleft to bind here at this receptor. And this can have a variety of effects when you have that. It could, for example, cause the channel open up and cause an EPSP. It could cause the channel to stay closed and prevent an EPSP. It could cause the channel open up and cause an IPSP. Or let's say that this is a typical inhibitory channel, it can prevent the inhibition. So there's a variety of ways and that a synapse can function to influence, and it depends on what's being released. We'll talk about those. But after the receptor activation we have here, we have to turn off that signal. So we turn on the faucet and we start to have the water flowing. But in order to have a signal, a communication system, we can't just leave the water running. We've got to be able to turn it off and turn it on when we want it, right? We have an action potential that we can initiate and terminate. We use rate to code the intensity of stimuli. How does that work in the chemical transmission that occurs at the synapse? Well, we have 4 different ways that we can inactivate the neurotransmission signal. First, and probably the simplest, is just simple diffusion. If you have a bunch of neurotransmitter in the synaptic cleft, it's going to want to diffuse away from that location, going from an area of higher concentration to an area of lower concentration. So simple diffusion can be sufficient as a mechanism to clear that out of the cleft. Another mechanism is that we can have enzymes that are located in the synaptic cleft, and these enzymes can break down the neurotransmitter. So here in this figure, what we have is acetylcholine, that neurotransmitter that Otto Lowy discovered in the heart. And we see that it's also used in the central nervous system. So here is the pathway in which we synthesize or make acetylcholine in the presynaptic terminal, where we're going to take different parts of it, transfer, and then we actually get acetylcholine. Once it's released into the synaptic cleft, we have something that's referred to as acetylcholine esterase, or an enzyme that breaks down acetylcholine. Once those 2 components are broken down, acetylcholine cannot influence the characteristics or the receptors postsynaptically. All these have to be reabsorbed presynaptically and go through this process again. So you have to make the acetylcholine has to be released, it has to be deactivated, has to be reabsorbed, and you have this continual kind of process. So those individual parts, the acetate and the choline, have to be reabsorbed so that then they can be combined to actually make acetylcholine again. But another way that we can deactivate the neurotransmitter outside the diffusion are the function of these enzymes that break down or change the neurotransmitter so they can stop binding postsynaptically. Another way that we can influence or change this function is reuptake. And I don't think I have a graph of that. But we can have, how we have sodium-potassium pumps that work to keep a higher concentration of sodium outside of the neuron, a higher concentration of potassium inside of the neuron. Well, we can actually have pumps that will actively transport or take dopamine, for example, or other neurotransmitters presynaptically. So there can be a reabsorption or reuptake. That will clear the neurotransmitter out. As soon as it is brought within the presynaptic membrane, it needs to be bottled up into our vesicles so that it can be released at a controlled time related to the occurrence of an action potential down there at the presynaptic membrane. Finally, glial cells. Some people view that glial cells reabsorb or absorb some of these neurotransmitters at the synaptic cleft. So astrocytes would be playing a large role there. So once we have the signal, we have the neurotransmitter synthesized, we have it stored, we have it released, it's activated, those receptors, we have to clear that signal away, and we can use diffusion, where it just simply goes from an area of higher concentration to lower concentration, so it gradually moves out of synaptic cleft. We can have enzymes functioning at the synapse to break down those neurotransmitters. Some of those neurotransmitters can be reuptaked or reabsorbed presynaptically and then packaged up so they can be released again. Or glial cells will absorb that neurotransmitter. So those are the 4 ways that we can deactivate the neurotransmitter. Now, when I took my first brain and behavior-like class, there weren't that many neurotransmitters, and there was a big focus on trying to hunt for more neurotransmitters. People were really excited. And what they had I think that drove some of the excitement is there were certain criteria that you had to establish to say that you found a neurotransmitter in the central nervous system. And they all depend on these stages of neurotransmission. So in order to make the claim that a chemical that you've discovered, just like Otto Lowy discovered acetylcholine, you have to demonstrate how that chemical is synthesized. So you have to be able to demonstrate that pathway. Does it involve the activation of the DNA, or can it be made on site through a couple of enzymatic reactions? What is that synthesis pathway? You also then have to demonstrate that it is released from some neuron. That may involve electrical stimulation of a neuron to get the generation of this, kind of like what Otto Lowy did with the accelerator, stimulating the accelerator or the vagus nerve in the heart. So when he stimulated that, he was able to demonstrate that a chemical was released. And then you have to, after you have demonstrated that it's released, you want to know that this chemical that you're studying, let's say acetylcholine, has an effect on the target that actually changes the function of that neuron. So you need to then take and apply that chemical that you've discovered postsynaptically and see that you can get either an excitatory postsynaptic response or an inhibitory postsynaptic response. You need to demonstrate that that chemical has an effect on neurotransmission. And then you have to demonstrate how is it deactivated. There's got to be some mechanism for removal of that chemical from the synapse. So you have to demonstrate that there's some deactivation pathway. And this set of criterion has been very effective in allowing researchers to identify neurotransmitters that are functioning to communicate information across the synapse. It has resulted in the discovery of an amazing variety of neurotransmitters. We'll talk about one. There are gas. Nitric oxide is an example of a neurotransmitter that we can figure out its synthesis, how it's released, how it has an effect on the target, and how it's deactivated. So this has been a really important tool in discovering or investigating how the central nervous system communicates information across the synapse. Now, to turn our attention to these neurotransmitters, we'll focus on two general classes. First class are referred to as small molecule neurotransmitters. And these building blocks are typically obtained from the diet, and they typically are fast-acting. A good example of one of these would be dopamine, or DA, is abbreviated. So dopamine is abbreviated DA. And here's the chemical structure of dopamine. So you get most of what you need to synthesize dopamine from your diet. And it doesn't require a great deal of synthesis from your DNA. These are very fast acting, so they'll be released, have an effect, and then they're cleared from the synapse. One of the things that I like to point out is the similarity between a drug that acts like dopamine, amphetamine, its chemical structure, versus dopamine's chemical structure. You can see that there is some clear similarity in the way this compound looks relative to the way dopamine looks. So that similarity is important for how amphetamine interacts with those receptors postsynaptically. But these fast-acting small molecule neurotransmitters also are synthesized, like I said, from the diet. And so we have this biochemical pathway where we'll eat tyrosine hydroxylase, that'll be from our diet, and then that, through an enzyme reaction, turn into L-DOPA. L-DOPA then will be, through another enzymatic reaction, turn into dopamine, and a further reaction of dopamine with an enzyme will translate to norepinephrine, and another enzyme reaction will turn it into epinephrine. So these series of steps allow us to synthesize different neurotransmitters, and depending on where this starts out, you can end with just dopamine, norepinephrine, or epinephrine, being released at the synapse. So there's a clear biochemical pathway for these small molecule neurotransmitters. Dopamine is involved in movement and also reward. And one of the centers that we talked about that was important for movement was the substantia nigra. And when we talked about the substantia nigra, we talked a little bit about the movie Awakenings because of its involvement in Parkinson's disease. And from the movie Awakenings, if you have a chance, check it out. It discusses individuals that had developed encephalitis, and that encephalitis damaged the substantia nigra, and they developed this very difficult paralysis where they couldn't move. And so the movie depicts them receiving a treatment, L-DOPA. And if you remember, L-DOPA is that second stage here where that now we're increasing L-DOPA, so we're giving them part of this biochemical pathway. That translates to potentially more dopamine, and that would allow for more communication within the basal ganglia. And overcoming some of that degeneration of those neurons that are releasing this pathway into the central nervous system. So by increasing L-DOPA, that precursor chemical to dopamine, you're essentially going to get more dopamine made and increase the function of the central nervous of the substantia nigra and the basal ganglia. And these people were able to come out of their paralysis that was the result of the Parkinson's disease that came from the encephalitis. So anyhow, it is a good movie, amazing at how well they were able to portray the disorder and the human component. So I encourage you to check out the movie. Another class of neurotransmitters are referred to as peptides. These now are typically slow-acting, very slow-acting, so they'll slowly be released. They are synthesized from activation of the DNA, and so they have to be synthesized, they have to be transported and released. These are also very slow to be replaced as well. So a good example of these peptides would be of the family opioids. So we have metenkephalon and leukonkephalon. These are involved with some of those natural pain inhibitors. We talked about the A-reactive ductal gray matter as being important in reducing pain related to species-typical behaviors. Well, we have these neurotransmitters that are located there, and their release, while slow to be replaced, they are effective in reducing pain associated with those acute situations. But because it takes so long to make them, they're not great for a chronic pain such as arthritis. So the synthesis of these neurotransmitters, these peptides, is telling in how they are good in an acute situation where pain may interfere with ongoing behavior, but the way that they're synthesized is informative based on more chronic, why we aren't able to handle pain with more chronic conditions such as arthritis. Again, peptides involve activation of the DNA. We transcribe that information from the DNA into messenger RNA. Messenger RNA then is translated into chains of amino acids, and long chains of amino acids turn into these peptides, and they are important for this example we're giving you here is important for reducing pain, and they would be located within the periapital ductal gray matter. So we can also talk about several receptors and give you an idea about how their effects can change the function of the central nervous system. one receptor, we have actually 3 classes of receptors, and these are typically thought to have different functions on the central nervous system. So we'll first talk about ionotropic receptors, and these are fairly straightforward, where a neurotransmitter will bind to a location, a binding site, and that will cause an ion channel to open up. So ionotropic, there will be a binding of the neurotransmitter, and then that will allow the ion to flow through that phospholipid bilayer. So as that neurotransmitter binds, all of a sudden you're going to allow certain ions to flow through. Now it really depends on where you're at and what the neurotransmitter is and what the ion is, because these ionotropic could be excitatory or they could be inhibitory. So they have a variety of effects that they could have. We could be letting some positive ions in, and we'd be producing an EPSB. We could be blocking some other ion. There's a variety of effects. What you should note about the ionotropic receptors is that once the binding occurs, it produces rapid changes, and then the transmitter will be released, and there's no lasting effects. So you can imagine these being important for those immediate kinds of reactions, being able to, if you're taking notes right now, using your hand or your fingers to direct the pencil. So how are you able to cause those to go, movements of the pencil to go in the correct direction? Well, you have to imagine that there are motor neurons, interneurons, as well as sensory neurons, all communicating information, and you have all the electrical signals going through the neurons, but within the synapses between those neurons, you have to get the signal across the synapse, have it its effect, remove it, and then move on so that you can now move in the other direction. So ionotropic receptors are going to be critical for that type of behavior when you need very rapid changes in information processing with very little lasting effects. So that's the function of the ionotropic receptors. Now, not all such circumstances call for that rapid, no lasting, very limited lasting effects. Metabotropic receptors now can have a little bit longer effects, and we have 2 types of metabotropic receptors. These metabotropic receptors can be coupled to an ion channel. One of them is coupled to an ion channel, and what happens is you have a neurotransmitter that will bind to a receptor site or a binding site, and then that will activate a G protein. And so after that G protein has been activated, what happens is an alpha subunit, as we can see here, will then come over and cause this channel to open up, this ion channel, allowing certain ions to flow through here and either produce an IPSP or an EPSP. So here we see a transmitter binds, it activates the G protein, then the G protein is activated, the subunit comes over here and opens that ion channel. Now this is going to take a little bit longer than the ionotropic receptors, but we can now, because this is an alpha subunit, instead of just directly activating the ion channel, we can see that this will become longer effects than just the ionotropic receptor can have on neural communication. So the changes are a little bit slower, but their effects are a little bit longer. So you might think of this as trying to keep in memory something somebody told you, a list of things that you need to go shopping, and you want to keep that in memory, so you want to have some of that information accessible, but you don't want to have to keep that for the rest of your life. So you can kind of see these as being those intermediate effects of some kind of stimulation. You can imagine that you get scared, and then after that frightful event, you're still, for a while, kind of heightened in your arousal with that. That would be another example of how there may be this slower changes but have a little bit long-term or shorter-term effects, longer than what we'd see with ionotropic. Finally, we have metabotropic receptors that are coupled to enzymes. So it's the same kind of thing where we have a neurotransmitter that was released and that binds to this site here on the receptor. And doing so, that activates the G protein. But now the G protein subunits are activated, and they come over and activate this enzyme that will trigger a second messenger. So this is another messaging system within this postsynaptic neuron that can then translate to activate DNA. That means you can build new ion channels. You can build more neurotransmitter. You can build other types of things that this neuron needs. So because you're activating the DNA. So this, while it may have very slow changes, it can have lasting effects. These can be represented in terms of the memories that you have that will last a lifetime. Can be related to the activity of these metabotropic receptors that are coupled to enzymes. So it will be very slow. It might not even change. It might not, during the event that you're learning, taking notes from this class, it might not be involved at all in changing the membrane potential, but you know what it does? It puts more ion channels in place so that the next time you're confronted with that information, you'll have more channels in place and you'll be more likely to respond than had you not had that experience. So these really are the basis of memory, these metabotropic enzyme-coupled receptors. Again, what happens is a neurotransmitter binds to the receptor. That activates the G protein we see here. The alpha subunit now comes over and activates this enzyme. After that enzyme is activated, it triggers a bunch of second messengers. And those second messengers will then activate the DNA so that there's an increased synthesis of certain proteins, maybe ion channels, maybe neurotransmitter, maybe a combination of both, so that the synapse is fundamentally different in its firing characteristics. So with that discussion about those metabotropic receptors and their impact on the long-term consequences of neurotransmission, we get to talk a little bit about synaptic learning. And really, I think this is one of those quite exciting areas for neuroscience, where we can see nice structural, functional changes that are clearly associated with the behavior changes we observe, and we can narrow down or at least develop these animal models that really let us gain insight to a deeper understanding of what are memories, where are they stored, what are the processes. So the eventual goal with that understanding is to develop better treatments. One of the disorders that currently confronts our society is Alzheimer's disease, and it's almost exclusively a memory disorder. It's a problem with these people generating new memories. And so when we try to study and understand memory, we can't always study it in people. We have to develop animal models. And one of them that was developed by Eric Kandel is the aplysia. And so here we see a picture of the aplysia. It's a large snail about the size of a softball that lives in tidal pools. And it is nice in that it's fairly easy to work with. The central nervous system is fairly simple, but also it learns from experience. It changes its behavior. And so Eric Kandel used those features to study what is happening at the synapse in an animal that's learned about its environment. And we'll talk about two forms of non-associative learning. If you're interested in this and you want to find a little bit more about associative learning, I teach a class, Psych 431, Neuropsychology. It's a lab class, so if you're in psychology, you may want to consider taking that. We get to dive deeper into associative learning and talking about those mechanisms that relate to that. But for this class, we'll just talk about non-associative learning. It's a form of learning, a change in behavior due to experience. And we'll talk about two forms, habituation and sensitization, and we'll see how that has been used to study this system. So let's get started with habituation. First of all, let's talk what habituation means in terms of behavior. So with the aplysia, it lives in these tidal pools close to the ocean, and it normally will like to breathe through a gill. So here we can see is the gill that's extended here, and that's how it likes to normally breathe. But oftentimes, because of where it's located, there'll be something that may try to attack it, and it will want to protect. It has fairly tough skin, but the gill is very sensitive to those attacks, so it will retract it. And even if you just squirt, because it's so sensitive, some water on the siphon, that's this portion over here, it will produce this nice kind of retraction trying to protect it. It doesn't know if that water is a bird, a gull, a seagull trying to peck at it, or what, and so it will retract that. It can still breathe. It has to breathe through the skin, and it's not its preferred mode of respiration. But we can see here that when we activate or touch the siphon, that gill will retract it to protect it. Now, since they live in tidal pools, that means the tide's going to come in and out, and they'll start to get used to the water stimulating the siphon. So if we continue to squirt the siphon, what happens is that it will no longer retract the gill or pull that gill in. And that's, by definition, behaviorally, habituation. We define habituation as a decrease in responsiveness due to continued stimulation. And we've had that experience all the time in our daily lives. For example, right now, you're probably not thinking about the way your foot feels in your shoe until I just mentioned it. Now you feel your foot in your shoe. That's an example, a great example of non-associative learning that's habituation, that you aren't aware of that until I draw your attention to that phenomenon. So in the aplysia here, we have a very simple behavior that we can produce that depends on experience. So you have to stimulate the siphon multiple times in order to see this habituation behavior. And then the question is, are there any changes in the aplysia's very simple nervous system that correlate or are associated with these behavioral changes? So how simple is the nervous system? Well, I'd say it's pretty simple. We have basically a sensory neuron, as we can see here, that's that is conducting information or collecting information from the siphon. So we have a sensory neuron that synapses directly on a motor neuron that goes out and will cause a gill muscle to contract. So that's very, very simple. And this is in your book as well. So if you want to read through it, in addition to what I'm saying, that might be beneficial. So the sensory neuron synapses on a motor neuron, and what we want to do is look at aplysia that have never had this habituation kind of experience with the water activating the siphon versus aplysia that have had this habituation process, and to see, are there any differences occurring right here at the synapse? And this figure comes right from your textbook, and what you see is there are changes that occur. And one of the things that we see in an animal that's had the this kind of habituation experience where we've squirted the siphon multiple times with water, we see that there's a decrease in the calcium influx that's occurring when an action potential travels down this sensory neuron. So if there's less calcium coming in, that means less of it's binding with the calmodulin. That means fewer of these vesicles are being released. That means less neurotransmitter is being released. That means there's less neurotransmitter binding postsynaptically. There's less channel activation. That means there's less EPSPs, excitatory postsynaptic potentials, being generated here at this membrane, and that if you have fewer EPSPs, that means you're less likely to generate an action potential in this motor neuron, and that decreases the tendency to withdraw that Gill response. So we can see this treatment, this behavioral change that we see is associated with a decrease in calcium entering the presynaptic membrane here, or this presynaptic neuron, this neuron. So there is a change we're seeing occurring at the synapse that seems to be related to this momentarily change in responsiveness to that Gill withdrawal response. Again, in animals that haven't gone through habituation, they show that there's a normal amount of calcium that enters, that binds with the vesicles, that releases the neurotransmitter, that neurotransmitter binds postsynaptic, that generates sufficient EPSPs to elicit an action potential that travels down that motor neuron and then causes that Gill to retract. So there's a change that's occurring here at those calcium channels that's not allowing as much calcium to flow in with the habituation. Now, another form of non-associative learning that we can see is called sensitization. Sensitization is also non-associative, so that means we're not trying to associate two things. It's just some change in behavior. Our definition of sensitization is an increase in responsiveness due to continued stimulation. So we see there's an increase in some type of response due to some type of sensitization. We can think back to or imagine what it's like for the aplesia there in those tidal pools. And you can imagine an episode where maybe a gull comes down and starts pecking at the bum of the aplesia. And so it's really worried about that Gill getting torn up. And so that initial kind of pinch to the bum from the gull may make it actually hyper-responsive. It can't tell if the water or it's getting attacked. I like to think back of when I was in graduate school, to give you an example of sensitization that occurred, where we were going out to the movie theater and we're going to go see the Blair Witch Project and the drive-in. And so we were pretty excited. You know, I don't know if you've seen it. I think it's on Netflix. At least it was. And we went to the theater. We saw it. We got pretty scared, pretty aroused. And on the drive home, we lived kind of in the country. And so as we were parking the car, we heard something rustle in the bushes. And we all kind of stopped and looked, and it was a squirrel, but the movie had sensitized or increased our responsiveness to other stimuli in the environment. Much like the aplesia, when it's attacked by a gull, now just a little splash of water on the siphon will cause it to withdraw its on the siphon will cause it to withdraw its Gill. So how do we stimulate that consistently in the lab? Well, we could try to consistently pinch, but that, people may pinch differently, and you don't know that you'll pinch always at the same time like a seagull would. So what we do is we apply a mild shock to the bum of the aplesia, and that is enough to stimulate this being attacked by a gull. It allows us to do it multiple times without damaging the skin and give a consistent stimulus. When we do that, now, if we squirt the water jet, we're able to cause a dramatic withdrawal of the Gill, and every little water jet will cause that to withdraw. We have basically a nice behavioral model of sensitization. The question is, are there neural changes at the synapse associated with this behavioral change. And again, this is what Kandel operated on, is the simplicity of the central nervous system, the clear evidence of this learning from experience, and trying to study are there changes that are occurring with this experience in the central nervous system. Now we have to increase the complexity of the central nervous system a bit here. We're going to, have you think about 3 different neurons instead of just 2. We still are going to be collecting information from the siphon of and with sensory neurons. We're still going to require the motor neuron to cause the gill to retract. But now we have an interneuron that's collecting information from the other parts of the body, such as the bum. So when we apply that mild electrical shock, what's going to happen is that interneuron is going to come and influence this functioning of the synapse between the sensory neuron and the motor neuron. So let's focus in on that synapse. Again, this figure is in your textbook. So here we have the interneuron. It's activated because we shocked the tail of the aplysia. We now are just going to apply a little bit of water to the siphon, and that's the sensory neuron here, and then we're going to be looking at what happens to the motor neuron. Since we activated the interneuron, we're going to release serotonin. Serotonin is going to be released here from the interneuron, and that's going to influence the functioning of the sensory neuron. Specifically, what it does is it activates a series of second messengers, or like neurotransmitters within the sensory neuron, that's going to influence the function of potassium channels. And so these potassium channels are not going to allow as much potassium to flow out. Now remember, what was the important role of potassium in the action potential? I'll give you a second here to go back and maybe look through your notes to see if you can answer that question. So what was the role of potassium in the action potential? What did it do? We talked about the different phases. What was potassium important for? Did you find it? If you're saying that it actually prolongs the action potential, or it's preventing it from leaving prolongs the action potential, in this case, then you're correct, because during the action potential, potassium terminates. Remember, potassium leaving decreases or gets that repolarization and hyperpolarization. So in this case, with the aplysia, if the serotonin is activating these second messengers, and these second messengers are preventing potassium from leaving, that means the action potential is going to reverberate around the sensory neuron much longer than normal. And if it continues to reverberate, we'll get more calcium influx. More calcium influx translates to more vesicles being docked and more neurotransmitter being released. More neurotransmitter being released translates to more binding, neurotransmitter binding, more EPSPs, and if we have more EPSPs occurring in a certain time frame, we increase the likelihood of generating an action potential down that motor neuron, increasing the likelihood that we'll withdraw that gill. So we can see that reducing potassium leaving those channels prolongs the action potential at the siphon, and this goes right back to that importance of understanding the action potential. So understanding how potassium and sodium function in the action potential and seeing that we can modify that activity and get a change in behavior, such as sensitization, demonstrates the importance of understanding at this molecular level. Now, these are, so if you were to do either habituation and sensitization in a day in the lab, you would see these changes occurring. So these are relatively short-term changes. What would happen if we brought the aplysia in and we squirted their siphon every day, or we shocked them every day for a week or 2 weeks? what would happen? Would there be some long-term changes that would occur within the central nervous system? Well, your book goes through a study, or describes a study that did just that. So under control conditions, you can see that we have this sensory neuron that has many synapses on a motor neuron here. So we can see that is what's happening in a control condition. But if you were to bring in an aplasia and squirt it every day on the siphon for several days, let's say a week, what you would see is there would be a structural change. There would be a reduction in the number of synapses here. And that's what you would see in an animal that has become habituated. However, what would happen if you were to continue to peck or apply shock to the bum of the aplasia across a week or so? You'd see an increase in the number of synapses across that motor neuron from the sensory neuron, increasing that neural communication. And how do you think? What do you think the mechanism of that structural change is? Activation of the DNA, involving building or removing certain amounts of that neuron, either activating to decrease the amount of membrane, those synapses, or acting to build more synapses and increase those synapses across there. And so our discussion of those different types of receptors illustrate the importance of being able to, depending on experience, activating the DNA and causing structural change. That structural change then will translate into more permanent behavioral change and provides evidence that the synapse is that really central portion, or plays a central role in forming these long-term memories. And being able to study this in the aplasia is wonderful, because we're dealing with neurotransmitters like serotonin and ions, potassium, that we see in humans, non-human primates, as well as in other organisms, fruit flies, and allows us to make these inferences about communicating information from one neuron to another neuron and develop animal models so that we can better understand the function of the system and develop better interventions when the system goes wrong, such as what we see with maybe Alzheimer's disease. Well, again, we covered a lot of ground, so I would hope that you would review this several times before attempting the lecture quiz of what content we talked about here. You should be taking notes from these, pausing it occasionally to make sure that you have all these components down, maybe rewinding. Also be taking notes out of your textbook, and then attempt those practice exams as an assessment of where you're standing and seeing how you're doing with this. Our next chapter is quite exciting. We get to talk about how drugs come to influence this functioning or this communication across the synapse. So until then, I'll say goodbye.